Welcome. In this lecture on aids to health, we would be talking about the concept of immunity, the various types of immunity, the local immunity, which is the non-specific immunity, as well as the specific immunity, and further the types of specific and non-specific immunity. To begin with, what is immunity? The idea of immunity is simply to protect our body from infection, which is identified by the body as non-self. What is self and what is non-self? Self is those things which are part of your body. However, non-self is any external thing that attacks your body. Now, immunity prevents or protects your body from those non-self elements. Now, when the pathogens enter the body, there could be various lines of defense. We call them as the first line of defense, the second line of defense, and the third line of defense. The idea of each of these is to fight with the disease. For example, I take a very simple case of skin. Now, skin protects the germ from entering in the body. If there is a wound or a cut, there is higher probability for the infection to enter. But in case of an uninterrupted skin, we could say that there is a very less chance that that pathogen or that germ can enter into the skin. So, skin provides a type of defense mechanism. To begin with, the non-specific immunity. The non-specific immunity is also called as the local immunity. This immunity could be classified as the external defense system and the internal defense system. Any external defense system is called as the first line of the defense. Extremely important, a very important question. The second is, what is the second line of defense? The internal defense system. The internal defense system includes WBC, macrophages, interferons. The first line of defense, which is the external defense system, could be either physical or chemical. When I say physical, skin, as I said, is the first line of defense. Here, cilia, now these cilia are present in the windpipe that sweep the bacteria into the throat. Mucous membrane present in the stomach is another an example of physical or the first line of defense. So, skin, mucous membrane, hair and cilia are classic examples of physical first line of defense. Under the chemical first line of defense, there is sweat glands, oil glands, tears, hydrochloric acid from the gastric juice, similarly bile from the hepatic juice are ones that destroy the bacteria that enter the stomach or the gut. The next is the second line of defense. The second line of defense is known as the internal defense system. This could be classified as WBC, macrophages and interferons. Let's understand each of this. WBC is basically the neutrophils that squeeze the wall of the capillary, and this is occurring by the process of diapesis. Now, when the diapesis occurs, it engulfs the pathogen inside it and destroys it. Pus, as we see as a secretion, is what? Pus is a secretion of those killed WBC, the damaged tissue, as well as the pathogen which is destroyed. The next is macrophages. Macrophages are large tissues which are released through the lymph, and these are released either through the lymph, the spleen or the linings of the blood vessel. They move to the site of infection and act in a similar way as WBC acts. The next is interferons. Interferons are chemicals which are made up by the dying cell. So the dying cell which was affected when a viral infection occurred would release certain chemicals. Now these chemicals would protect the other cells from getting infected by that viral, and this is what is known as interferon. It is interfering and therefore it is called as interferon. So this local or non-specific defense system, which includes the first line of defense and the second line of defense, why is it important? It is important because as soon as germ enters, it gets in contact with either the first or the second line of defense. This is non-specific in nature and it is not dependent on your previous exposure to the infection. It starts defending the body as soon as the infection or the germ is seen. The second is the specific immunity. The specific immunity immunity is known as the third line of defense. Now this specific immunity is generated through antibodies. Antibodies develop in response to antigens. Antigens are any foreign molecules that enter into the body. They could be from the cell wall of the bacteria, from the coat of the virus. They enter into our body and antibodies start to fight with those antigens. Now this is what is called as the specific immunity. Now specific immunity can be classified into two forms. It could be an immunity which is by birth. We call it as innate or inborn immunity. The other immunity is through the one which we acquire. Therefore it is called as acquired or adapted. The next important thing under innate or inborn specific immunity is, it could be specific or non-specific in nature. When I say specific, it is targeting to a specific organ, a specific cell. When it is non-specific, it is applicable to whole of the body. So innate or naturally acquired acquired immunity is important. Now how does a person acquire immunity? So this is innate, that is by birth. The next is acquired. Now the acquired could be active or passive. Active immunity which is acquired is due to a previous infection that a person might had, or the antigen that enters into the body either naturally or artificially. Now active immunity, how would it generate? Through natural way, either by infection, artificially through the vaccination. Both of them generate long-lasting immunity, right? The acquired immunity can also be passive in nature. Passive means the body has the ready-made antibodies which are there. Now these ready-made antibodies, how do they come in the body? They can be obtained naturally again or artificially. Naturally, how they are obtained? Naturally, they can pass from mother to child through the placenta. Artificially, they can be produced in a horse body, let's say against the snake poison, and once an individual gets a snake bite, that poison, that venom which is created from that clot could be artificially injected into the person. We would understand how antivenoms work in a while to understand this better, how the antivenoms work. The idea here is, to summarize again, the immunity can be local or non-specific and specific in nature. The non-specific one could be external, which is the first line of defense. Hair, skin. Then under the chemicals we have the secretions through sweat glands, sebaceous glands as an example of the first line of defense. The second line of defense could be WBC, interferons and macrophages. The third line of defense, which is the specific immunity, could be either inborn, which is acquired by an individual at the time the child is born, or which is not acquired, which is by birth, but the other form is acquired, which could be acquired naturally by the child from the mother, or it could be given artificially as and when required by the body. Now, how does this antigen antibody work? As I said, antibodies are created in reflects to the antigens, the foreign complexes that enter into the body. Now antibodies belong to immunoglobulins, and these immunoglobulins are produced by the plasma cells which are derived from the lymphocytes. So these antibodies are derived from the plasma cells which are produced by the plasma cells which are derived by the lymphocytes, and they respond in light to the antigen. So there is an antigen antibody complex formation that occurs, and this is what is called as antigen antibody complex. The binding could occur by three ways. One is the lysis of the cell, where the foreign molecule or the foreign body that enters is destroyed. So this is called as cell lysis. The next is phagocytosis, where the foreign body is engulfed within the cell. We call this as phagocytosis. So this could be either by neutralization, where the viral binding sites and the bacterial coats are actually neutralized. There could be a precipitation of the antigen, or there could be agglutination. Agglutination actually means the various particles sticking together. So they bind together and form one. And those are the ways through which there could be binding of antigen to antibody, and this inactivates the antigen. So this would be a form of response by the antibody. Now in certain cases we have seen that the antibody response does not work. For example, in severe diseases like HIV, this response does not work. Why does it happen? The simple logic here is HIV attacks the T lymphocytes. Now as the T lymphocyte, there are two lymphocytes, T lymphocytes and B lymphocytes. When T lymphocytes are attacked, it stimulates B lymphocytes to produce the immunity, but since the T lymphocytes are destroyed, B lymphocytes are not able to produce the antibody, and therefore the immunity towards that infection is not generated, and this leads to weakening of the immune response. Now two important terms under this is vaccination and immunization. Immunization simply is to make a body immune for a certain disease. Now how can it be immune? It could be by providing the weakened germs or the killed germs in one form or the other. The idea is to resist the infection from affecting the individual. What is vaccination? The vaccination is a method through which you introduce the vaccine into a body of an individual, and this is to make a person resistant to the disease. There are four types of vaccines that we talk about. One is the killed vaccine. Again, very important. Killed vaccine, good example is Salk vaccine for polio, TAB for typhoid, and then we also have the rabies vaccine, which is a killed form of vaccine. The next is living or weakened vaccine. Living or weakened vaccine includes measles, mumps, there is freeze and dry of tuberculosis, BCG are examples of living and weakened vaccines. The next is fully virulent vaccines which are there, and then surface vaccine. Surface vaccine, a good example is influenza vaccine. So influenza vaccine by purified surface molecules is a good example of surface viruses. And then there are also toxoids. What are toxoids? Toxoids are actually the elements which are killed, and they create a coat, and this coat acts as a surface one for the others and prevent the infection from entering. A good example of toxoids are tetanus toxoid, TT as it is called as. So tetanus vaccine comes as tetanus toxoid, TT, and then we have diphtheria toxoids. So these two are given in the form of toxoid. Now who discovered vaccine is an important question. Jenner discovered the concept of vaccine. He was the one who coined the term vaccine as well as vaccination. He, in one of his studies, found out that a person who was infected by cowpox was immune to smallpox. This study was done where the virus of the smallpox to a person infected by cowpox was handed to Sarah Nimmels, who was one of the experimentees, and there he realized that this person is now immune to the smallpox because had a previous exposure of cowpox, and this led to generation of the first vaccination which was done by Edward Jenner, and therefore Edward Jenner is the one who is attributed to the idea of vaccine and vaccination. Toxoid, as I said, is what? It is the extract of the toxin, and this toxin is secreted by the bacteria. Now this toxin which is secreted is mixed with formalin. Now once it is mixed with formalin, it reacts in a certain way that it becomes harmless, but it retains the properties of an antibody, and therefore, as I said, it creates a cover which is used, and therefore it is used as a vaccine. So we call it as a toxoid vaccine, which is very important. Tetanus and diphtheria are form of toxoid vaccines. Now if we focus on the National Immunization Schedule of India for infants at birth, what are the vaccines which are given? So BCG, hepatitis, oral polio vaccine 1, 2, 3, DPT, which is diphtheria, pertussis and tetanus. Then we have hepatitis B, measles and vitamin A, the first dose of it are given for infants. Then for children, which is from 16 to 24 months, it is the booster for DPT, measles, oral polio, and a vaccine for Japanese encephalitis if required, if in an area which is endemic or prone to Kala Azar. In those areas we have this Japanese encephalitis which is given. The next is vitamin A dose and DPT booster doses at later years if as and when required. So there are various other formulations which are given. The next important topic that we would understand is what is antitoxins and serums. Antitoxin, as the name suggests, is working against the toxin which is released, so we call this as antitoxin. The antidote of a snake bite is what? It is known as antivenin. Now how this antivenin is created is important. That snake venom is given in a very low dose to either a horse or a rabbit. Now when it is given to horse or a rabbit, the blood sample is extracted, the blood is allowed to clot. Now once the blood is allowed to clot, the serum which is left after the clotting has that antivenin property, and that serum is stored, and this is used as an antidote in a case of snake bite for human beings is as and when required. So antitoxins have a specific property to neutralize a specific toxin for which it is made. Snake bite, snake venom, antidote, antivenoms are one example. Other are tetanus, again made by similar methods. Diphtheria, toxoid, again made by similar methods. The next is what is serum. Serum is a whitish yellow part of the blood which is left behind when RBC have been removed. Now, once a blood clotting takes place, the RBC, the fibrogen, the blood plasma removes, the yellowish part, the whitish part which is left behind is what is called as blood serum. Now, this serum is used as an inoculation, is injected into the safe amounts as and when required and worked as a serum. Serum Institute of India has been working for creating these serums and vaccinations based on it. The next is antiseptics and disinfectants. Very important, the difference between those. Antiseptics are mild. They are used for wounds, injuries. They don't harm an individual. Disinfectants are strong. They harm the skin. They are used for household cleaning, hospitals, kitchens. Antiseptics, Savlon, Dettol, those are the brand names, but what is the composition is important. Iodine, hydrogen peroxide, benzoic acid, boric acid are common constituents of antiseptics. Under disinfectants, phenol, sodium hypochlorite, hydrogen peroxide in higher quantity and concentrated form, potassium permanganate are example of disinfectants. They cannot be used for wound healing. So there is a clear difference between the two. The next is understanding antibiotics. Okay, antibiotics are what? When an individual is affected by a bacteria, we use antibiotics. By a viral, we use antivirals. Now, how are these antibiotics prepared? Now, antibiotics are prepared. The term was coined by Selman Walkman, and this was in 1942. The antibiotics initially were derived from bacteria, yeast, and various other components. The first example of antibiotic which was discovered was by Alexander Fleming, and this was penicillin. Now, penicillin, how it was discovered is interesting. In London hospital, in the agar jelly growth which was seen in the petri dish, it was seen that the agar plate was contaminated with Penicillium notatum, and this when contaminated, each jelly, each bacteria started to have a jelly around it. Now this jelly growth stopped other bacteria. So this was because of the penicillin. Therefore, penicillin was used as an antibiotic. The first treatment where it was used was in the disease of gonorrhea, which is a STD, a sexually transmitted disease. And then penicillin acted as a wonder drug. After that, there were series of antibiotics in the form of tetracycline, penicillin, grisofulvin, which were released. Now here we have certain examples of antibiotics which are bacteria borne, which are made from bacteria. So polymyxin, bacteriacin, gentamicin, erythromycin are other examples which are of the streptococcus family, streptomycin family. Similarly, from the fungus, we do have antibiotic creations which are grisofulvin, penicillin and cephalosporin. So these three antibiotics are created from fungi. Now, antibiotics, sorry, it is antibiotics, not antibodies. Okay, antibiotics could be narrow spectrum or broad spectrum. Broad spectrum means it has a capability to treat variety of diseases. However, narrow spectrum means it is meant for specific targeted diseases. Narrow spectrum antibiotics include penicillin. It also includes streptomycin, erythromycin, syndamycin, metronidazole. However, broad spectrum antibiotics are those which are used to treat variety of diseases. Amoxicillin, augmentin, azithromycin, tetracycline, moxifloxacin are broad spectrum. Aureomycin is another broad spectrum antibiotic. Now, within this, there is another class of drug which is known as sulfonamide. Sulfonamide is basically a composition from sulfa. So they are called as sulfa drugs. They were initially used to treat meningitis, inflammation of meningitis, or it was also used to treat urinary infection, urinary infectory tract. Later on, there were various other disorders through which sulfonamide was used. However, it is important to note that if this drug goes in overdose, it could lead to fever, rashes, malfunctioning of kidney. So, prescribed doses are again important for any antibiotics, specifically those from the sulfonamide family. From the sulfonamide family, it could be sulfonadiazine, it could be sulfenoxazole or sulfonamide itself. The next is, when we talk about health, various organizations and their role is important. The local organization, the municipal organizations cater to the health services, the cleanliness, the safe drinking water, good medical centers, training of midwives. We have better family planning centers, better primary health care centers. The various common diseases that we address in India are malaria. So there is a national eradication malaria control program. Similarly, vector-borne control program. There is pulse polio, cholera control, filariasis control, Institute for Communicable Disease, which is working in line with those. The next is two important international organizations. One is Red Cross, the other is WHO. Red Cross has a symbol of red cross, a cross of red color, and this indicates a voluntary organization established by Henry Dunant, and this was in line of the memoir that he had written, a memory of Solferino. Solferino was the battlefield where the French and the Austrians were actually at fight, and there were more than 40,000 casualties which were witnessed by Henry Dunant, and this led him to develop an organization which came in the name of Red Cross later on. It was accepted by UN convention to establish this, and this was founded in 1864. Later on in 1920, it came and established in India with its center. The idea of Red Cross is to provide blood donation during the wartime, to provide mother and child care centers, to train the midwives, to provide immediate assistance for refugees, for migrants, to people with distress, to whom ambulance services are required and normal emergency services cannot be reached on those areas. In those areas, Red Cross does a remarkable work. The next is World Health Organization. The work of World Health Organization has a broad spectrum. The headquarters are located in Geneva, Switzerland, and this was established in 1948. The idea is to understand the current epidemic scenario, the future epidemics, the support and the preventive measures, the proper vaccination to each countries, the international nomenclature for various conventions, the control of epidemic, the workforce that could be sent for the control, and a detailed description of the various diseases, their cause, their treatment and symptomology. So those are some of the important things that come under the ambit of World Health Organization. The role of World Health Organization is more broad in contrast to the Red Cross, which has a very specific and focused vision, mainly in times of emergency situation, crisis situation, when countries are at war, when military actually requires support, ambulance services are needed. In those time, in case of humanitarian relief efforts, disaster that has occurred in a specific area, Red Cross plays a very important role. So this was an important topic. I hope you were able to extract the key points. Just to summarize, the first, second and third line of defenses are very important. What is antibody? What is antigen antibody interaction? Antibiotics, broad and narrow spectrum antibiotics are some of the key things. The examples of broad and narrow spectrum antibiotics are again important from this chapter.